Hello and welcome to BioRules video lessons. Today we are talking about physiology, and in physiology we'll talk about digestion. So you probably have lots of information about digestion. Probably you like to digest because you probably like food, because eating food is something that's amazing and we love to do it. So what is going to be this topic about digestion? So if you're going to be talking about digestion, we have to talk about food. So let's get this cheeseburger here as an example of food. The cheeseburger actually is made by several different components. You have the bread, you have the sauce, the onions, the tomato, the lettuce, the cheese, the meat, and another bread in the bottom. So if you get the bread, most of it's going to be starch. You have starch in the onion, you have starch in the tomato, you have cellulose in the lettuce, another polysaccharide. But then we have lipids and proteins mainly. In the cheese, we have lots of proteins and lipids in the meat. And so basically different part of food here are made of different stuff. And the main stuff here in the food is going to be starch or cellulose, which are polysaccharides. It's going to be lipids, which are the fats, and it's going to be the proteins. So when we think about starch, we should remember this kind of molecule. This here is an amylopectin. It's one of the starches that is basically a bunch of glucose glued together. Now it's time for you to revise your biochemistry. If you think about proteins, they are a bunch of amino acids bonded together, making a three-dimensional shape. If you think about lipids, they can be cholesterol, but what we're going to be playing with here in the digestive system is going to be the triglycerides. It's a glycerol and fatty acids. So these here are the three main macromolecules in our diets. We eat to be able to get carbohydrates, we eat to be able to get proteins, and we eat to be able to get fats. Okay, cellulose is also part of the diet, but we don't eat to get cellulose. We get cellulose, which helps the digestion process, which helps the digestive system, but it's not something that we are actually going to be using in our life. Okay, so first thing, to digest is to break down something. So digestion is the breakdown of molecule. So in our case, the digestion of food is going to be the breakdown of large food molecules into small food molecules. So we need to break these very large molecules, because large molecules are very difficult to be absorbed by the epithelium of our intestine. So our intestine is not capable of absorbing starch or cellulose or proteins or carbohydrates, complex carbohydrates, and so on. But it is possible for us to break these big molecules into smaller molecules. So food macromolecules, basically we are going to be talking about those three categories. Polysaccharides, it can also be disaccharides, monosaccharides, but the big ones that really need digestion is going to be the polysaccharides that we will end up breaking in the end into monosaccharides. Polypeptides, which are the proteins, which will in the end be broken down into amino acids. And triglycerides, that in the end is going to be broken down into glycerol and fatty acid. Those are the big food macromolecules. Okay? Sucrose actually is not a polysaccharide, it's a disaccharide, but it also requires digestion. So I'm going to be using it here as an example. There's an enzyme called sucrase that we use to break sucrose into a glucose and a fructose. Yay! So then there they are, it's broken. So this process, if you remember in biochemistry, is the hydrolysis. We put water, we have an enzyme, and that makes the breakdown of a disaccharide into monosaccharide. So sucrose, which is the table sugar, the Brazilian sugar, is broken down into glucose and fructose, and then our body can absorb glucose and fructose. Okay, just another example here. This is a piece of a polypeptide is broken down into amino acids. So basically here you have NCC, double O, this would be double OH. So this is an amino acid, and in here we have two other amino acids. So basically this reaction, we broke 3 peptides into an amino acid and G-peptide. So this is the beginning of the end of digestion of protein, because in the end it has to be broken down into tiny little amino acids. So G-peptide, or in this case the tripeptide, is going to be broken into amino acids. And the glycerol is the same idea. You have a triglyceride here with glycerol and 3 fatty acids. We break it down into glycerol and 3 fatty acids. For that we use an enzyme called lipase. So this is the digestion of the lipids. Quite simple, quite straightforward. Okay, but for breaking down these chemicals to make the chemical reaction of hydrolysis, we need enzymes. So why do we need that? Also remembering biochemistry, enzymes, they speed up chemical reactions. So they make the chemical reaction go faster. But in the case of digestion, they speed up chemical reactions so much that without them, it would be as if you didn't have any breakdown at all. So if you don't have the enzyme, very few molecules are going to go through the process of hydrolysis. They will end up entering, leaving your digestive system, and you're not going to be able to make use of it. So we need enzymes to speed up the breakdown of these molecules, but speed up so much that actually it makes possible for our body to absorb it in the time that the food is inside of our body. Otherwise, it will take way too long. Okay? We need to know some enzymes in the digestive system. I'm going to be showing you many here. You need to know, for the IB diploma exams, you need to know a carbohydrates, a protease, and a lipase. So I'm going to show you some stuff here, and I'm going to show you where they are. So there is one enzyme called salivary amylase, which optimum pH for function is 7. It's produced by your salivary glands. It's thrown in your mouth, and it breaks starch into maltose. Starch is a bunch of glucose together. Maltose is 2 glucoses together. So it's almost finishing to digest the starch. It's there. It works there in your mouth. Okay. One more example: pepsin. Pepsin is a protease. Of the sins, they are protease. The optimum pH is 2. It is produced by your stomach. So your stomach is a gland because it produces chemicals for you, produces enzymes for you as well. So it's going to be breaking large polypeptides into smaller polypeptides. And it's going to be working at the stomach itself. One more example, a lipase, a pancreatic lipase, which optimum pH is going to be 8. And it's produced by the pancreas. It breaks the triglycerides into glycerol and fatty acid, and it's going to be acting in your small intestine. To be specific, it's going to be thrown in the duodenum, the beginning of the small intestine. It's going to be working the duodenum, jejunum, and the ileum, the remaining of the small intestine. Those are 3 enzymes for you to know that they are going to be working into our body. Okay. Where they're going to be working. So amylase, they work over there, pepsin in the stomach, lipase in the small intestine, but you have another enzyme called trypsin that works in the intestine, but produced by the pancreas as well. Okay, you have pancreatic amylase that also works in the small intestine, produced by the pancreas as well. And we have other enzymes that are not in here. Even the small intestine also produce its own enzymes. So some of the small intestine enzymes, they are produced in the pancreas and they work on the small intestine, but some are produced by the small intestine itself. Okay, so just a few enzymes over there. And to make a table for you to try to remember the name, the pH, the optimum pH, the source, the substrate, and the products of the enzymes, let's put over there. Salivary amylase, pH 7, produces salivary glands, breaks starch into maltose. Pancreatic amylase, optimum pH 8, because the small intestine pH is 8. Produced by the pancreas, it breaks the starch into maltose as well. Maltase, pH 8, produced by the pancreas, breaks maltose into glucose. Lipase, which is pancreatic lipase, optimum pH 8, produced by the pancreas, works in the small intestine, breaks triglycerides into glycerol and fatty acids. Pepsin, not Coca-Cola-sin, optimum pH is 2, produced by the stomach, works in the stomach, and breaks large polypeptides into smaller polypeptides. Trypsin, produced by the pancreas, optimum pH 8, because it works in the small intestine, it breaks the small polypeptides into even smaller polypeptides. Okay? And biosalts, they are not enzymes. So you know some enzymes now here, we have listed here some for you. But biosalts, also known as bili, they are not enzymes. So what are they? Biosalts are emulsificators or emulsifiers. They are like soaps. They work as amphipathic molecules. They have a hydrophilic part and a hydrophobic part, like soaps do. And the function of the biosalts is to isolate big blobs of fat that, because of being apolar in a liquid environment of the food in the intestine, they get all glued together, and the enzymes don't manage to work very well if you have a big blob because of surface area to volume ratio. What the biosalts are going to be doing is to separate these big blobs of fats into small blobs of fat, so the enzymes are going to be able to work, so the lipase can do its job. Okay? It's produced by the liver, it works in the small intestine, it's accumulated in the gallbladder. Okay? So liver produces, accumulates in the gallbladder, and is thrown into the small intestine. The idea is to separate hydrophobic fat lumps so the lipase can work, the small pieces. This is the function of the biosalts, which are not enzymes. Okay? So, short and sweet, you have now some information about digestion and enzymes and the need for digestive enzymes to break down food. The idea is for our food to be in molecules small enough so our digestive system can absorb it into our bloodstream. Now you have a little quiz to do, and I'll see you in class. Bye bye.